Physics colloquium. It is pleasure to introduce our speaker today, Professor Peter Hore, from University of Oxford. He spent most of his life in Department of Chemistry at Oxford University. First as a student between 1973 to 1980. Then he moved to as a postdoc into University of Groningen, and then he came back to University of Oxford, and now he is working as a professor at Department of Chemistry, University of Oxford. He worked on various topics around NMR and nuclear spin electron and nuclear spin effect on chemical reactivity, as field known as spin chemistry. This includes spin hyperpolarization, protein structure and folding, photosynthetic energy conversion, and NMR methodology. Since 2005, he is working on trying to unravel the biophysical mechanisms that allow migratory songbirds to detect direction of Earth's magnetic field as an aid to navigation. So today he is going to talk mostly on that topic, and the title of the talk is Quantum Birds, a Magnetic Compass Mechanism. So once again I welcome Professor Peter Hore.
Good afternoon. Thank you, and thank you for the invitation. I'm delighted to be here. I'm having a great time, and I'm very grateful for your hospitality. Small songbirds like the European robin and Africa and back again. Quite remarkable distances. How do these birds manage not to get lost? Well, they can use the sun and the stars to get a sense of direction. They can also recognize odors and landmarks. But it's also clear that they have a magnetic compass. That is, they can detect the direction of the Earth's magnetic field and use that to help them to find their way. And that's what I want to talk about today. How does this magnetic compass work? We've known for 60 years now that the birds have a magnetic compass. We still don't really know what the mechanism is. So first of all, how do we know that birds have a magnetic compass? Well, to do experiments on wild birds, one needs to bring them into the laboratory. And the assay that is widely used by many groups around the world is called the Emlen funnel method. So you put a bird inside a funnel-shaped orientation cage, shown in this cartoon, with a lid on top so it can't escape. The cages are cylindrical, and so the only directional information the birds get inside these cages is the magnetic field that you subject them to. And during the migratory seasons, in the spring and the autumn, when the birds are motivated to migrate, in the evenings, when the free birds would be preparing to fly through the night, caged birds exhibit a behavior known as migratory restlessness. They become agitated, and inside the Emlen funnel cages, they hop around in an attempt to escape. And the direction in which they jump inside the cages is the direction in which they would fly if they were released. So it's a very well-controlled assay for migratory orientation. And as shown in this cartoon, the way that you record the bird's orientational preferences is to line the sloping sides of the cage with paper that is sensitive to the scratches that the birds make with their claws as they attempt to escape. And from this cartoon, this particular bird evidently was oriented on average towards the northeast. So the way this is done in the laboratory of my collaborator, Henrik Mauritzen, at the University of Oldenburg in northern Germany, is to test 9 birds at a time, one in each of these Emlen funnels, for an hour at dusk. So each cage has a translucent lid, so light can get in from above, but the birds can't see out. So once again, the only directional information they get is from the magnetic field. And that field is supplied by a 2-meter cube of coils, 4 in each of the 3 dimensions. And by carefully controlling the currents through these coils, one can produce a magnetic field very homogeneous in the center where the 9 Emlyn funnels are positioned. So all you need to do to show that the birds have a magnetic compass and that they're using it is to repeat the experiment with a rotated magnetic field, say by 120 degrees. And then if the birds hop in a different direction, rotated by roughly 120 degrees, then you know that they are perceiving and using the magnetic field to orient. And it's experiments of this sort that in the 1960s first convinced people that European robins really do have a magnetic compass. And so this testing facility, the coils you see here, are in a carefully electromagnetically shielded chamber that allows the Earth's magnetic field in, but not time-dependent magnetic fields. So any electromagnetic noise which might interfere with the bird's ability to orient is excluded by the shielding. And there are 4 such chambers inside this testing facility. So this sometimes we call the DARPA house. It was built with funding from the American agency DARPA, and it's made entirely of non-magnetic materials and is used for these behavioral assays, testing magnetic orientation. So what I want to talk to you about is what is currently the leading hypothesis for the mechanism of this magnetic compass, which is that there are chemical reactions inside the birds' bodies which are sensitive to the direction of the Earth's magnetic field. So the basic idea is quite straightforward. You have a set of chemical reactants which get converted into products via a short-lived intermediate molecule. And that intermediate can do one of 2 things. It can either return to the reactants on the left or go forward to the products on the right. So all you need for this to be the basis of a compass is that the branching ratio, the probability that the intermediate goes left or right, depends on the magnetic field in which you do the reaction. And so then the yield of the reaction product will encode the magnetic field strength and direction. So all the bird needs to do is to move around in the Earth's magnetic field and register the change in the yield of this reactant product. And that will give it information from which it can deduce the direction of the magnetic field and therefore in which direction it wants to fly. But as soon as you start thinking about this, you realize that it's a ridiculous idea, because the energy of interaction of the Earth's magnetic field with a molecule is about 1,000,000 times smaller than the thermal energy, Boltzmann's constant times temperature. Absolutely tiny magnetic interaction. And so there's no way you would think that such a weak interaction could affect the rate of a chemical reaction or the position of a chemical equilibrium. Nevertheless, there is a mechanism that is sensitive to such weak magnetic interactions. And this is a well-established mechanism. It's been known for 40 or more years, and there have been many laboratory studies of a special type of chemical reaction which proceeds through radical pair intermediates that does show effects of weak magnetic fields. So I'm going to spend the first few minutes outlining this mechanism. So imagine that the reactants are 2 molecules, A and B, or perhaps 2 parts of the same molecule. And they are diamagnetic. That is, they have closed electronic shells. All the electrons are paired up. There is no net magnetic moment. These are not paramagnetic molecules. And then, if you shine light on A and B, let's suppose that triggers an electron transfer. So it jumps from A onto B. And that will form a pair of radicals. So molecules that have an odd number of electrons. They each have one unpaired electron. So these are paramagnetic. They have magnetic moments. And since we're going to be dealing with organic molecules in biological systems, there are no heavy atoms, and so that electron transfer process conserves spin angular momentum. So because the reactants were overall in a spin singlet state with no net spin angular momentum, so the initial state of this radical pair is also singlet, which we think of, roughly speaking, as the two electrons, one in each radical, are anti-parallel, as indicated by these red arrows. So we start off in a singlet state. But the radical pair doesn't stay there for very long, because the singlet state is not stationary in the quantum mechanical sense. Rather, it is a coherent superposition of the spin eigenstates of this radical pair. And internal to the radicals, there are magnetic interactions between the electron spins and the spins of magnetic nuclei, like hydrogen and nitrogen. These are called hyperfine interactions, and they break the symmetry between these two radicals, and they drive coherent interconversion of the singlet radical pair and the corresponding triplet state, which, roughly speaking, has parallel electron spins, one in each radical, shown by the blue arrows. So the radical pair oscillates coherently, quantum mechanically, between its singlet and triplet states. And you can show that with a simple calculation. Time-dependent quantum mechanics. Choose a spin Hamiltonian that contains a few typical hyperfine interactions of the electron spins with magnetic nuclear spins. Start it off in a singlet state and then calculate here over a period of 2 microseconds the probability that that radical pair is in a singlet state as a function of time. And it oscillates. What you're seeing here are quantum beats. And the frequencies are of the order of a few tens of megahertz, reflecting the strength of those internal magnetic interactions, the hyperfine couplings. So this is in the absence of an applied magnetic field. If you repeat the calculation, but with a 50 microtesla magnetic field present, so that's roughly the strength of the Earth's magnetic field, then the quantum beats change. Now you can see there is an oscillation superimposed on the previous one with a period of around 0.7 microseconds. And that reflects the Zeeman interaction of the electron spins with this 50 microtesla magnetic field with a frequency of 1.4 megahertz. So what's happening here is that the frequencies of these quantum beats are just given by the separations of the energy levels that are in coherent superposition. And by applying the magnetic field, you move those energy levels around and split previously degenerate energy levels, and so give rise to additional frequencies for the quantum beats. And if you rotate that magnetic field, say by 90 degrees, the spin dynamics change again. And that's because the hyperfine interactions are anisotropic. The radicals we're going to be talking about don't have high symmetry. They have low symmetry, and that's reflected in those hyperfine interactions, so it matters what the direction of the magnetic field is relative to the radicals themselves. And that directional dependence, of course, is crucial for a magnetic compass sensor. So, even though the interaction with the Earth's magnetic field is 1,000,000 times smaller than the thermal energy, it can affect the spin dynamics of this system on a time scale of the order of a microsecond. So that thermodynamic argument is not applicable because the spins are not at equilibrium. And so to complete the story, you just need a bit more chemistry. So the singlet radical pair, let's say, recombines here in a spin selective fashion back to the reactants. The corresponding process for the triplet would be spin forbidden. Remember, the electron transfers must conserve spin. And then we imagine that the triplet state can react forward to form a signaling state. And so at any instant, the probability that the radical pair reacts to the left or the right depends on the probability that it is either a singlet or a triplet. And that probability depends on the direction of the magnetic field. So by the time all of the radical pairs have reacted one way or the other, the final yield of this signaling state will contain information on the direction of the field in which the reaction took place. And so in principle, that could be a chemical compass sensor. So that's the theory. So to make that perhaps slightly more intelligible, there is a mechanical analog. So imagine that a chemical reaction is like tipping over a heavy stone block. Now to do that, you would need to input a certain amount of energy to raise the center of gravity of the block to get it to tip over. And so imagining that a chemical reaction could be affected by the Earth's magnetic field is analogous to imagining that a fly bumping into the side of this block would affect the rate at which it tips over. That's clearly not going to happen. The energy involved is far, far too small. However, if you look at the same thing a slightly different way, suppose you've put the energy in already to get the block into this intermediate state, where it's poised on its edge, a highly non-equilibrium state. It could easily fall forward or back. And now a fly falling on one edge could make all the difference to whether it goes on to form the product or back to form the reactants. So, tiny amounts of energy, if applied to suitable, highly non-equilibrium states, can have profound effects. And that's essentially what the radical pair mechanism is in this context. So as I say, it's an established mechanism. We know that it works in the laboratory, at least for magnetic fields considerably stronger than the Earth's magnetic field. So the big question is, could it operate inside a bird's body? And is it sensitive enough to be the basis of a compass sensor? Okay, well, to start answering that question, a few facts about the bird's magnetic compass. If you test a bird in the Emlyn funnels, in the Earth's magnetic field, in the northern hemisphere in the spring, when it's motivated to fly north, then it experiences a magnetic field shown by this black arrow. The horizontal component points north, the vertical component points down. If then you repeat that orientation test in a magnetic field in which the horizontal component has been inverted, then, not surprisingly perhaps, the birds fly, or try to fly, hop in the opposite direction in their cages. Of course, a magnetized compass needle of the sort we used to use when we were hiking before we had magnetometers on our phones would also point south under those circumstances. More surprisingly, though, if instead you invert the vertical component of the Earth's field, which you would think would have no directional information, then the birds still go south, even though the horizontal component is still pointing north. And they only go in the right direction when you exactly invert the field, both horizontal and vertical components, so that it points to the right and up instead of to the left and down. So this is not like a compass needle of the sort that we would use. It's not a polarity compass, it's what's known as an inclination or axial compass. And so whatever the mechanism is, it needs to reflect that property. Also, it's light dependent. If you test the birds in the Emlyn funnels under blue light, then they can orient perfectly well in the migratory seasons in the Earth's magnetic field. If instead you use green, yellow, or red light, or total darkness, they jump in random directions. They can't use their magnetic compass. They need the blue wavelengths from visible light, from the electromagnetic spectrum, in order for the compass to work. Okay, so what do we know about the location of the sensors? Well, they're in the bird's eyes, which perhaps is the most obvious place for a light-dependent sensor. More specifically, they are in the bird's retinas, the thin layer of cells at the back of the eyes responsible for vision. So a bird's retina is a very crowded layer of cells, all of which are involved in turning the energy of light into signals that allows the birds to see. And it seems most likely that the magnetic sensors are in the double cone photoreceptor cells, right at the back of the retina. So birds have 4 different cone cells tuned to different colors. They have rod cells for night vision that are not color sensitive. And they also have these double cones, which is the most common type of photoreceptor cells in the avian retina, and their function is not well understood. And it seems that the most likely location for the magnetoreceptors is in these double cones. And within the double cones, the most likely molecule that could play host to this radical pair chemistry is a protein called cryptochrome 4a. And we know for sure that it is in the double cones and also in the red single cones. So cryptochromes were only discovered in the early 1990s, and by the end of that decade it was clear that they formed radical pairs when you irradiate them with blue light. That was discovered 25 years ago, and since then there's been no other candidate suggested as the magnetoreceptor molecule in birds. It's the only known vertebrate photoreceptor that forms radical pairs when you shine blue light on it. Doesn't mean it's the right molecule, but it's the only one we have at the moment. And the evidence suggests that it is indeed involved. So, in the middle of cryptochrome, shown here on the left-hand side, is a molecule labeled FAD. That's a flavin adenine dinucleotide. It's non-covalently bound in the middle of the protein. It's a yellow compound closely related to vitamin B2, and it absorbs in the blue, roughly the same wavelengths that the birds need for their magnetic compass. And it's linked to the surface of the protein on the right-hand side of this slide by a chain of 4 tryptophan amino acids, shown in green, A, B, C, and D. And what happens when the flavin absorbs a blue photon is that it triggers a series of electron transfers within the protein. The first one from tryptophan A onto the photoexcited flavin, and then an electron jumps from B to fill the hole in A, and then from C to B and D to C. So this tryptophan tetrad behaves like a molecular wire inside the protein, allowing an electron to flow from D all the way to the flavin, a distance of about 2.5 nanometers, forming a radical pair. One radical being the flavin, which has gained an electron, the other radical being the terminal tryptophan at position D, which has lost an electron. And we can do experiments on the purified cryptochrome 4a proteins and show that the subsequent photochemistry of these radicals does indeed respond to applied magnetic fields. So it looks like this molecule is suitable for the purpose of magnetoreception. Its photochemistry is magnetically sensitive. And we know that the electrons come all the way along this chain because we can prepare mutants of this protein in which each of these 4 tryptophans, one at a time, has been replaced by a different amino acid that doesn't do electron transfer. So we can block the electron transfer chain at each of the 4 positions, and in that way be sure that the electron is coming all the way from the end of the chain. So we have a molecule that's in the right place and seems to have the right properties to be a magnetic sensor. So to summarize this part of the talk with a cartoon, we have the ground state of the protein shown here by the green blob, a shape supposed to represent the conformation of the protein. Excite that with blue light, that forms a singlet radical pair. Then comes the quantum mechanics, the coherent interconversion of singlet and triplet states. Then the singlet radical pair can undergo a back electron transfer. The electron on the flavin can go back along the chain to the terminal tryptophan, returning the protein to its resting state, or the radical pair can go forward to form a signaling state. And there will be competition between those 2 pathways, a competition that is regulated by the magnetic field in which the reaction takes place, the Earth's magnetic field, and dependent on its direction. So the signaling state inherits magnetic information from the short-lived radical pair. The signaling state, as indicated by the different shape of the yellow blob, has a different conformation. There's a change in the shape of the protein, and that signaling state could be much longer lived than the radical pair. And its quantum yield, as I say, depends on the magnetic field. And from there, everything happens in the dark and much more slowly. The first event will be a biochemical signal transduction cascade. So having got this conformational change in the cryptochrome that affects its ability to bind to other proteins, whose identity we're trying to discover, that leads to a signaling cascade with biochemical amplification. Neurotransmitter release allows transmission of information between cells in the retina, and then ultimately the ganglion cells at the base of the retina generate action potentials that go along the optic nerve to the brain, where that magnetic information is integrated with directional information from other cues. And then the bird decides which way to fly. So that's the idea. So how do we test it? Well, so we've done the chemistry and physics now. Now we're coming on to the biology. But that's okay, because the biology is really quite simple. So what we want is a behavioral test. Something we can do with the Emlyn funnel assay and test to see whether this really is a radical pair mechanism, and if it is, have we got the right molecule? Well, you'd think that would be easy. All you have to do is to breed a bird that has no cryptochrome 4a in its eyes, and then see whether it can still orient. Well, unfortunately, that's not possible at the moment. So these are wild birds. They cannot be bred in captivity, and so one cannot do genetic manipulation. Very difficult, even to knock down cryptochrome in a bird's retina. So for the time being, that kind of experiment can't be done. So birds are not fruit flies. Drosophila, of course, one can do wonderful things with transgenic flies. So we tried that. We wanted to use Drosophila as a model organism for studying avian magnetoreception. Well, flies are not birds, but they have cryptochrome, and some people believe that they use the Earth's magnetic field to navigate. And there have been some high-profile publications reporting magnetically sensitive behaviors in Drosophila. So we thought we would simply replicate some of those studies and then start studying transgenic flies in different magnetic field conditions to try and learn something about how it might work in the birds. And so to cut that long story short, we tested more than 100,000 flies, and we couldn't find any statistically significant, reproducible, magnetically sensitive behaviors. So it's back to the birds. We can't use flies as a model system, at least not in our hands. So it turns out there is another magnetic field effect on migratory birds that we could use to get insight into the mechanism. And that's the effect of time-dependent magnetic fields superimposed on the Earth's magnetic field. So this was first done 20 years ago by Thorsten Ritz and his colleagues in Frankfurt. And what they did was to test birds in the Earth's magnetic field, in the Emlyn funnels, subject to radio frequency fields in the range up to about 10 megahertz, with intensities up to 100 nanotesla. So very much weaker than the Earth's magnetic field and time-dependent. And what they found was that the birds could no longer use their magnetic compass. They jumped in random directions. And since then, half a dozen groups around the world have been able to replicate that effect. So unlike a number of things in this area, it does seem to be independently reproducible. It looks like it's a real effect. And it happens whether the radio frequency field is a single frequency, monochromatic, or whether it's a broadband noise modulated field. And in different people's hands, 4 different migratory species have shown this effect, and 1 non-migratory species, the zebrafinch. That was with a different assay, because zebrafinches don't migrate and don't show migratory restlessness. So it looks like a robust behavior. So we wanted to extend those experiments to learn more about how this compass might work. So we tested Eurasian blackcaps in higher frequency time-dependent fields. And the first 2 measurements were either centered around 5 megahertz or 80 megahertz. So these are noise modulated broadband radio frequency fields with bandwidths of around 10 megahertz. And at both of those frequencies, with a field of the order of 10 nanotesla, the birds were unable to use their magnetic compass. They were desperate to migrate. It was the migratory season. Without the radio frequency field, they were well oriented. With this much weaker radio frequency field, they jumped in random directions. We then tested 2 higher frequencies centered at 140 and 240 megahertz with the same kind of field strength as at the 2 lower frequencies. And these higher frequencies had no effect on the birds. They could still orient perfectly well. So it looked like there was a cutoff somewhere between 80 and 140 megahertz. Anything below that did affect the birds, anything above that didn't. So this is a kind of spectrum, and you're probably thinking that it's the worst 4 points over a frequency range of 250 megahertz. Well, you have to realize that each of these 4 points took 1 year to get the data. You need to test several birds several times throughout the 2 migratory seasons, spring and autumn. And you need to do that in at least 2 migratory periods to be sure that the effect is real. So you don't get many points per PhD. Okay, so how can we understand this? Why would there be a cutoff seemingly between 80 and 140 megahertz? What does that tell us about the mechanism? Well, the key to it seems to be an experiment that has been done in laboratories on radical pair reactions for the last 40 years, and it's known as RIDMA, Reaction Yield Detected Magnetic Resonance. So if you are a magnetic resonance spectroscopist, or you know one, you'll know that we love acronyms. So here's another one. So the idea is that you do a radical pair reaction in a static magnetic field and a time-dependent magnetic field. And you choose the frequency of the time-dependent field to be in resonance with a transition, a magnetic resonance transition in the radicals. Or to put it in other words, you choose the radio frequency to match one of the frequencies in the quantum beats. And if you do that, then you will move spin population from one energy level to another, and you will affect the coherence of pairs of spin energy levels. And so you will alter these quantum beats if you do that magnetic resonance experiment on radical pairs in the laboratory. And that will change the yield of the reaction product. And you can detect that in other ways. The reaction products typically are long-lived, and so you can just record at leisure the yield of the reaction products as a function of the frequency of the radio frequency field. So that's a well-established form of spectroscopy. So could that happen in the birds? So if the same thing happens to the radical pairs in the birds' retinas, then you will be changing the amount of the signaling state. And that could corrupt the information that the bird needs to use its magnetic compass. And that could account for the disorientation. So that's the hypothesis. Okay, so what kind of frequencies are we talking about? Why should tens and hundreds of megahertz be the appropriate ones? Well, that all comes down to these internal magnetic interactions, the hyperfine couplings. So shown in the middle here, with the colored blobs, these are representations of the molecular orbitals in the flavin radical and the tryptophan radical, the molecular orbitals that contain the unpaired electrons. And you can see the unpaired electrons are delocalized. In the flavin, over the 3 aromatic rings of that tricyclic ring system, and in the tryptophan on the right-hand side, over the 6-membered and the 5-membered rings. So these are aromatic compounds with delocalized electrons. And so that means that the unpaired electrons in these radicals have spin density spread over the radicals. So we can expect basically that all of the magnetic nuclei shown in these 2 structures will interact with the electron spins. And if you count them up, there are 4 nitrogens and 11 hydrogens in the flavin, 2 nitrogens, 10 hydrogens in the tryptophan, a total of 27 nuclear spins interacting with the electron spins. So it's a very large spin system. Easy to calculate just how big it is. All you need to know is that for every hydrogen you have there, you double the number of energy levels, and for every nitrogen you triple the number of energy levels. And so the total quickly mounts up. So we have a very large spin system with 27 of these hyperfine interactions. Okay, so shown here on the left-hand side is schematically those energy levels. I've only shown, of course, a tiny fraction of the total. And so what frequencies could we expect would affect the spin dynamics? Well, it's quite clear that the maximum frequency would correspond to this red arrow. That's the frequency that connects the lowest energy level with the highest one. So that's the maximum frequency that could have a resonance effect. So any frequency lower than that, the blue arrows, for example, they will be in resonance with some transition because there are so many energy levels. Any frequency lower than the red one will be in resonance with something, and so should have an effect, especially because we're using broadband radio frequency fields with a spread of frequencies, not just a single frequency. And any frequency bigger than the red one, the green arrows, would not be in resonance with anything, and so should have no effect at all on the spins. So the red frequency should be the cutoff. Anything below that, in principle, could upset the birds. Anything above that frequency should have no effect. So all we need to do is to calculate that red frequency. So you take your enormous spin system, 2 electrons and 27 nuclear spins, you form a matrix representation of that spin Hamiltonian, and you diagonalize that matrix to find the eigenvalues. And you take the difference between the maximum and the minimum eigenvalue, and that will give you the frequency of the red arrow. And we can do that because we know what all these hyperfine interactions are. These can be calculated reasonably accurately using molecular orbital theory. So you put all that into your computer, calculate the maximum and the minimum eigenvalue. Take the difference, and it comes out quite remarkably, I think, at 115 megahertz. Right between the blue frequencies that do affect the birds and the green ones that don't. So it looks like we could be on the right track. We seem to be able to predict this threshold frequency, the cutoff in the behavioral experiments. So if you want another acronym, it could be ADMR, Animal Detected Magnetic Resonance. We're doing a magnetic resonance experiment on the radical pairs in the bird's eyes, and we're using the bird's behavior to read out the fact that we've done a magnetic resonance transition. So it's not often you can sit at your desk at a computer, diagonalize a matrix, and predict how an animal will behave. But it looks like that's what's happening here. Okay, but maybe this is a coincidence. What we would really like to do is to change that cutoff frequency in a predictable way, and then repeat the experiment and see if the birds then are disoriented at different frequencies. Well, you will have noticed that I haven't talked about carbon yet. So 99% of carbon atoms are carbon-12, which doesn't have a magnetic moment. Its nucleus has an even number of protons and neutrons. They're all paired up, and there's no net nuclear magnetic moment. 1% of carbon is carbon-13 with an extra neutron, and that does have a magnetic moment, but it's only 1%, and so it's not going to have a major effect. But what if you could enrich the flavin radical inside the cryptochrome, in the photoreceptor cells, in the bird's retina, in the bird's eye, with carbon-13? And so instead of having 1% carbon-13, you have 100% carbon-13 at every carbon in the flavin, in the cryptochrome. What would that do? Well, that would give you many more hyperfine interactions. There are quite a lot of carbons in the flavin molecule. And they're close to where the unpaired electron is. And so they will be strong hyperfine interactions. And so now there will be many more energy levels spread over a wider frequency range. So the red arrow will be longer. There should be a higher frequency cutoff. And if you do that calculation, the prediction is the cutoff is close to 200 megahertz. So that now 140 megahertz, which previously had no effect on the birds on the left, now, if we're on the right track, that should cause disorientation. Well, we don't know the answer to that yet. Those experiments are in progress. As I speak, we're in the migratory season at the moment in Oldenburg, in northern Germany. And so maybe in a year or so's time, we'll have the results to know whether that works as we hope or not. And that, I think, if it does work, would be the most convincing evidence so far that this really is the way the birds orient magnetically. Okay, so now a question that is often asked is, what do the birds actually perceive? How do they know what the direction of the magnetic field is? Well, we don't know the answer to that. So this bit of the talk is going to be pure science fiction. But we know quite a lot about cryptochrome now and how it should respond to different magnetic field conditions. So we can calculate what a bird might perceive as it moves around in the Earth's magnetic field. So this is based on a huge number of assumptions, but it's possible that the birds literally see the Earth's magnetic field. Perhaps at night, when the cone cells are not active because of the low light levels, maybe the visual reception pathway is taken over for magnetoreception, so that the birds can use the cone cells literally to see the Earth's magnetic field. And so that's the idea behind this little video. So there's a compass rose at the top right, which shows you that the bird is currently looking due south. Well, it's not really a bird, it's a drone. A couple of years ago, we hired a drone and flew it above the Oxfordshire countryside close to where I live. We flew it in a circle with a video camera hanging underneath it. And then superimposed on this video, we put the pattern that we think that the birds might derive from a cryptochrome-based magnetic compass. And so when I set this going, what you will see is that the compass rotates as the bird looks around, and then as it approaches the northerly direction, you'll see a very distinct pattern emerge. And when that's dead center in the field of vision, that's when the bird is looking along the north-south axis. So here we are. The bird is starting to look around, southwest now. You're starting to see this pattern appear in blue and pink. We've just gone through west, now it's northwest. We're coming around to north, and there's this very distinctive cross shape. And now it's dead center in the field of vision. And that was when the drone was facing towards due north, magnetic north. And so, well, a bit of whimsy perhaps, certainly science fiction, but the birds must perceive something that gives them a sense of direction. Okay, to summarize what I've done so far, the radical pair in cryptochrome 4a does seem to be fit for purpose as a magnetic compass sensor. We know that from the experiments on the purified proteins. Its magnetic orientation can be affected by radio frequency fields, and that's consistent with a radical pair mechanism. And the frequency dependence so far is consistent with this flavin-containing radical pair in cryptochrome. And we can test that with carbon-13 birds. So that's all very well, but it would be very nice if there were some applications of all of this. Well, there are organic light-emitting diodes, as I'm sure you all know, in the displays of your mobile phones. There are closely related molecules that show magnetic field effects. So the output of an OLED of the right sort can be tuned by applying a magnetic field. And it turns out that the mechanism of those magnetic field effects on electron-hole pairs in these organic semiconductors is basically the same physics as the radical pairs I've been describing to you. So if we could really understand what happens in the bird's eyes, maybe we could adapt that to make magnetic sensors, which might have favorable properties in terms of not containing heavy atoms, which might be toxic or rare or expensive. And so this might be a source of magnetic sensing devices for human use, but not based on the limited number of amino acids that nature has given to us, but based on the almost limitless ingenuity of organic chemists to make molecules with specified properties, magnetic properties, to show desirable magnetic field effects for sensor design. The other potential application is in conservation of wild animals, in particular threatened migratory birds, threatened by human activity, habitat destruction, global warming. Typically, the way people try to conserve these threatened species is to move them to a nicer location. The trouble is, migratory birds are such good navigators, they just fly back again. So if we could really understand how they navigate and how they define home, then maybe we could fool them to stay in the safer environment long enough to raise young who would then treat that new safe environment as their home, so that when they migrated, they would come back to their safe new home. So finally, I would like to acknowledge all the people that have been contributing to this work over the years. I've tried to put the names of all the graduate students and postdocs on the relevant slides that I've shown you. I just want to mention briefly the more senior collaborators shown here. In particular, Henrik Mauritsson, biologist in Oldenburg, and Christiana Timmel and Stuart Mackenzie, chemists in Oxford. I'm very grateful to them, all of the postdocs and graduate students, and also to the European Research Council that's funded this research over the last 5 years. So if you wondered what birds think about when they migrate, well, it's not satellite navigation, sat-nav. They don't have GPS. Chemical navigation. So maybe it's chem-nav. I think we'll know in a few years whether that's the case. Anyway, thank you for listening.
The floor is open for questions.
Thank you for a very interesting talk. So I was wondering how there is directional sensitivity in the molecule. It's just nitrogens. There doesn't seem to be any spin-orbit coupling. So why is it sensitive to the direction of the magnetic field?
The spin density above and below the plane, so it's essentially p orbitals on the nitrogen. So those nitrogen hyperfine interactions are highly anisotropic. The component perpendicular to the plane is large. The component in the plane is essentially zero. So it's basically there's a node in the molecular orbital in the plane of the flavin. And you put your finger on it, it's those nitrogens in the middle ring of the flavin that dominate the anisotropy of the effect.
Fantastic talk. So, a quick question. How do you actually achieve carbon-13 enrichment?
I was hoping someone would ask. We feed the birds carbon-13 riboflavin. So birds, like humans, can't make flavins. They don't have the enzymes. And yeah, so that's what we hope will work.
Thank you.
Would climate change affect the sensors or the microreception?
Sorry, climate.
The climate change doesn't affect it?
The climate, well, so overcast skies don't matter. So the birds fly at a height of 1 or 2 kilometres typically, so they're above the cloud level. Starlight seems to be enough. They probably don't fly on rainy nights. Temperature. Climate change. Oh, I beg your pardon. Sorry, I missed that. Yeah, well, yes, it seems. I mean, the populations of migratory birds are dropping, and that could be for a variety of reasons. Climate change, I think, is quite possible to be one of the contributing factors. Yes?
What are the challenges using in this system? Appears to be very robust. So, what are the challenges in using this knowledge for quantum computing?
Sorry, the challenges for
Using this system in quantum computing.
in quantum computing, limited, I think. So some people have got quite enthusiastic about if there are mechanisms here that preserve the spin coherence, say for a millisecond. I mean, if that really happened and we could understand how it happened, then that principle could be useful for preserving coherence in quantum computers with very different technology. I can't think of a mechanism that would allow spin coherence to last longer than about a microsecond. So I think there are probably very limited lessons here for quantum computing.
One more you showed in one slide that with change in both the components of magnetic field, when the field is reversed.
When the Earth's magnetic field flips. That's a good question. I mean, if that happened instantaneously, of course, the birds wouldn't notice. However, it takes of the order of 10,000 years. And we don't know. I mean, there's no record of mass extinctions of birds during those periods in the distant past when the Earth's magnetic field has flipped. So my guess is they just switch off their magnetic compass sense and rely on other sources of information. I mean, certainly during the period of the magnetic field reversal, the Earth's magnetic field is highly irregular and locally, so would provide very little information, I think, useful to the birds. Yeah.
Hi. Hello. Hi. Yeah. So I read an experiment where they were breeding two different types of birds, and interestingly what happened was that the offsprings were inheriting the path from A to B from the other side and B to A.
I don't think I'm familiar with that.
But can you, in principle, breed two different kind of birds to see like how do the offsprings go?
Yep, sorry, I don't follow the question.
Okay, the question is
offspring of a bird somehow inherits the GPS map of the parent.
Oh yeah, sorry, yes, I mean, absolutely.
Yeah,
I beg your pardon.
It inherits the GPS map of both the father side and the mother side, like something.
Yeah, so the compass sense is indeed inherited. So the small birds flying alone without experienced older birds can navigate, from example, from Scandinavia to North Africa. So, I mean, that's very clear. The map sense that would allow them to do true navigation, so they would need to know where they are as well as which direction to travel. The map sense is learned. So indeed. But I mean, the problem is then that, you know, the birds in a new location, if they're relocated, would need to adapt genetically to that in order to pass on appropriate information to the offspring.
A lot of magnetic field. The electrons shield the magnetic field experienced by the nucleus. How come in these radicals the magnetic field experienced by the nucleus is a lot, or the magnetic field is very less, like 50 microns? And among spectrometers, we lay a lot of more Tesla.
Okay, but so if the question is, how can a magnetic field so much weaker than the hyperfine interactions have an effect? Is that true?
Here the weak magnetic field is...
Okay, well, so the Earth's magnetic field is roughly 50 microtesla. So the Larmor frequency in that field is 1.4 megahertz. Okay, so a period of 700 nanoseconds. So as long as the spin coherence lasts at least that long, there is time for the Earth's magnetic field to affect the spin state of the radical pair. So you just need coherence that has a lifetime of the order of the Larmor period. So the fact that it is an order of magnitude or more smaller than the internal interactions, in fact, is irrelevant. And you need the internal interactions anyway. So if the two radicals had no hyperfine interactions, then there would be no effect of a weak magnetic field. So you need something to break the symmetry. I mean, I'm not sure, does that answer your question? Okay.
Thank you for the talk, Professor. I wanted to add on to the question about directionality. So at the level of a single molecule, you said that there's a sense of directionality, but I would assume in the eyes of the birds, there's a lot of molecules and they'd be randomly oriented. So would there not be some sort of averaging effect there that...
Yeah, that's a good point. So, I mean, very probably all the double cone photoreceptor cells have cryptochromes, multiple cryptochromes. So then, by comparing signal to the retina, because of its curvature, that would allow the bird to work out the direction of the magnetic field. Indeed, there should be some averaging over those multiple receptors. The resolution, the angular resolution of the compass, doesn't need to be nearly as good as vision. Plus or minus a few degrees is probably good enough. So you can average signals over adjacent cells in the retina. Also, the time resolution doesn't have to be as fast as vision. So you could integrate over a period perhaps of a minute. If it took that long for a bird to get a compass bearing, that would certainly be good enough if it's flying through the night and just needs to update its bearing every hour or so. So for sure there will be averaging of all sorts there.
But within the cell, then all the molecules would be oriented the same?
Within the cell, all the molecules should be oriented exactly in the same way. And that's one of the reasons we think the photoreceptor cells are a good location, because they contain oriented membranes in which the opsin photoreceptor molecules are embedded. The cryptochromes are not membrane proteins, so they won't be in the membranes, but they could be associated with membrane proteins like the opsins. And that could be the way that all the cryptochromes in a cell are identically oriented and immobilized. Indeed.
In the video which you showed, for that to happen, there should be a magnetic lens to image that, right?
A magnetic lens?
Yeah, like to resolve this magnetic field, there should be a magnetic lens to resolve that field, right, in his vision.
So I think that probably the way to think about it is that receptors in different positions in the retina are pointing in different directions, and so they have different sensitivity to the magnetic field. So as the bird moves around, so do these sensors, and just with vision you see different things as you look in different directions. So you're moving relative to the magnetic field, and this changes the pattern. I think that's probably a better way to think about it than as a lens. I don't think there's any focusing
Yeah, like
there.
I was wondering, like since there is no resolving happening in the eye, so basically nothing is being focused on retina. So I was wondering if the background changes, the intensity of the background.
So there will be a background, because even without a magnetic field, there will be radical pairs formed and signaling state formed, so there will be signaling going on even without the magnetic field. And then superimposed on that there will be a modulation coming from the magnetic field effect. And so probably that constant background is filtered out in some way that we don't understand, to leave the information that is actually useful for direction finding.
Thank you.
Okay.
Thank you once again. So now we invite our HOD Professor P.N. Samush to give a moment over to the speaker.
Oh, gosh. Thank you very much. Thank you.
Thank you.